2 minutes to your slides while I introduce you.
I shared my slide.
Okay, perfect. Looks good. All right. Welcome everyone to our September episode of the GIS speaker series. Very proud to have Young Seok Ju as our invited speaker today. Young Seok is an associate professor at the Graduate School of Medical Sciences and Engineering at KAIST, the Korea Advanced Institute of Science and Technology. And many of you may be familiar with KAIST. It's really one of the foremost genomics and other research institutes in the world. And he's not just a researcher, he's also founder and CEO of Genome Insight Incorporated in Korea. And he's an associate professor at KAIST at the moment. Before this, a very prestigious lineage, he was a postdoctoral fellow at the Cancer Genome Project at Sanger, which, as some of you may know, is where a lot of the research and a lot of what we know today about genetics of cancer really comes from the Sanger. Okay, so he was at the place where, he was in the room where it all began. And he's done postdoctoral research at the Life Science Institute at Mitochondrial in Korea, which is one of the foremost whole genome or DNA sequencing companies in the world. So, and today he's going to talk to you about human genetics. Looking forward to your talk, Young Seok.
Thank you very much, Chayam. So I'm very glad, and it's my great pleasure to have a seminar in GIS. I hope to visit Singapore, but unfortunately, this is an online seminar. Whatever, so I'd like to start, and today the title is a Clonal Dynamics in Early Human Embryogenesis from Somatic Mutation. It's quite a long title, but my lab is looking at somatic mutations. So here is a mother cell, and here is cell division. And two daughter cells are very similar to the mother cell, but actually their genomes are not completely same, because there are some kind of errors during DNA replication or whatever, or there must be the impact of external mutagenesis. So the genomes of two daughter cells are not always similar, or there are some kind of somatic mutation in the daughter cell's genome. So the consequences of the somatic mutations are some diseases like cancer or rare diseases or whatever. But we can conduct a lineage tracing using genome sequences of daughter cells, because the daughter cells' genomes are not identical now. So the primary concept is we can trace the impact of daughter cells using somatic mutations. So probably it is much more familiar to you if I show the examples from cancers. So for example, in multi-region sequencing, we can sequence many different parts of the tumors and metastasize the tumor tissues. And using those somatic mutations, we can draw the phylogenetic tree of the tumors, and from this phylogenetic tree, we can understand the tumor clonal evolution in the tumor development of the patient. Or in the longitudinal sequencing, by sampling multiple tissues from different time points in the clinical courses, also we can trace the clonal evolution in the tumor tissue. So we can, this is the concept of the clonal, the lineage tracing using somatic mutation. So again, this is the when we do this work in the tumorigenesis, For example, let's say this is the most recent tumor ancestral cell, and let's say if there are two clones, for example, two subclones, then we can trace the proportion of each subclones in different tissues by using of somatic mutations. It is very clear. Now, our idea is we can apply the similar approach in human development. So for example, let's say these are two cells at two cell stage embryo, and each cell, each lineage, each cell has its own somatic mutation during the first cell division. Then by the mutation, we can trace the impact or the contribution of each lineage in many different tissues, because those progeny have their own specific somatic mutation. So here, what is required is that the mutation rate in early human embryogenesis or in the first cell division should be higher than one mutation per daughter cell per cell division in the embryogenesis. So that is the necessary condition. So during my postdoctoral research, I tried to estimate the mutation rate in early human embryogenesis. It was around 2015, and as you can see here, at that time, from the TCGA or ICG's International Cancer Genome Consortium or the Cancer Genome Atlas, they produced many tumor and normal whole genome sequences from many different tissue types, tumor types. So during that time, during my postdoctoral research, I reanalyzed those tumor and normal whole genome sequences in a totally different way. Let me explain you briefly. So let's say these are two cells at the two cell stage embryo, and each cell, let's say each cell acquired the specific mutation, early embryo mutation. Then we can distinguish two lineages by the somatic mutations. And in the polyclonal tissues, like in blood, those the lineages are admixed together, and they will show the somatic mosaicism. So this is the example of somatic mosaicism. So from the blood whole genome sequences, from the ICGC, International Cancer Genome Project, we found a mutation here. So this is here is mutation from the whole genome sequencing and also the targeted amplicon sequencing for the same region. So the mutation intensity is not 50%. Variant fraction is much lower than 50%, but it's not a sequencing artifact. So we thought that it is the somatic mosaicism in the polyclonal blood. What if we see the genome sequences from the matched tumor? So the chance is 50-50, right? The tumors can be derived from the first lineage, second lineage. And if the tumor was derived from the second lineage, all the tumor cells will have the specific mutation for the lineage 2. And as we can see here in the tumor genome sequences, we can see the mutations are much in higher intensity, or mutations are there in the clonally there. So we can say by combining two the analysis, we can say these are early embryonic mutation, or a mutation is in mosaicism in the polyclonal blood, and it is clonally present in the tumor tissues. So from the around 250 tumor blood whole genome sequences in my analysis, my calculation was the mutation rate was around 2.5 per daughter cell per early cell division, although the variation in individual variation is unknown, but the mutation rate is higher than 1. So we felt that we can try the tracking of early lineages using whole genome sequencing of the clones. So we can conduct this kind of we can apply the same the approach in human development. So for that kind of project, or application of similar project in the similar approach in human development, there should be the two necessary conditions should be met. So one is mutations should be accumulated in the first cell division. And at the time, the estimate rate was 2.5 per cell division, although its inter-individual variation is uncertain. Anyway, so it is over than 1, higher than 1, we can try the project. And the first step of the project should be the detection of the early embryonic mutations. So we termed it as the capture phase. And then after we identify the early embryonic mutation or EEMs, then we should track those EEMs in many different tissues in an individual, which was termed as the recapture phase. So let me start from the detection of early embryonic mutations in human individuals, or let me, this is the capture phase. So this is the conceptual, the explanation of the capture phase. This is for the initial detection of early embryonic mutation from an individual body. So from worm autopsies, I will tell you later why worm autopsies are necessary. But from autopsy samples, we can collect many cells, let's say 100 cells, for example, from many different anatomical locations, from leg or from arm or from liver, from brain or whatever. So we can collect many cells from many different anatomical locations, and then we can conduct whole genome sequencing of each single cells. And what we expect is we can identify thousands of somatic mutations per each single cell. Okay? And if the mutations are acquired early in embryogenesis, those mutations should be shared by multiple cells. So for example, when we analyze the thousands of mutations from each single cell, some mutations will be shared by multiple cells. And let's say those 5 cells share mutation A, and those 3 cells share mutation B, then we can reconstruct this kind of the early cellular phylogenies using somatic mutation. So for example, those 5 clones are progeny from this lineage, and those 3 clones are the lineage, the progeny from this lineage. So this is the very brief concept of the capture phase, or how we detected of early embryonic mutations. But there is a technical challenge. So that is how we can conduct whole genome sequencing of single genomes or single cells. So this part is quite challenging, because the most straightforward way for the single genome sequencing is actually whole genome amplification. So from each single cell, each human single cell, the amount of DNA, genomic DNA, is around 6.6 picogram. So it is too low. It's too small amount. So it is too small to conduct the whole genome sequencing. So for conducting whole genome sequencing, the genomes should be somehow amplified. So if we use whole genome amplification technique like the Malbec or MDA or whatever the kit, then we can technically conduct the whole genome sequencing. But because the genome amplification steps are error prone, so we will find so many false positive mutations, and more importantly, so many false negative mutations. So that means, so we miss so many the early embryonic mutation in many clones. So the analysis is not very robust. So we concluded that whole genome amplification method is not very useful for our analysis. So the alternative way is using the clonal expansion. So we can collect single cell, actually live single cell, and then we can amplify those single cells in vitro in the clonal expansion. So single cell to 1,000 to 10,000 K cells. Then from those, the colony, single cell driven colony, we can extract the genomic DNA higher than the amount will be the higher than 1 nanogram or 10 nanogram. So from that amount of the DNA, we can conduct whole genome sequencing without the in-vitro whole genome amplification. So the advantage of the clonal expansion followed by whole genome sequencing is that they provide very clean genome sequences. But the drawback is the single cell amplification step is costly, slow, and labor intensive. And the clonal expansion method is only applicable for live and proliferating cells. If cells are dead, we cannot apply this method. So that is why we need warm autopsy, or autopsies should be conducted immediately after death, hopefully in 24 hours of the death. And also our technique is only applicable for proliferating cells. So for example, neurons, because they are not proliferating, they are differentiated. So our method cannot be applied to the kind of the cell types. So anyway, so we tried 7 warm autopsies. The first 2 was kind of our first training set, and actually we chat. So this okay, let's try this chat. So from 5 individuals, we collected around 300 clones living the single cells, and we generated the clones, the whole genome, the single cell driven colonies from those individuals. And we conducted whole genome sequencing of those around 300 clones. Okay. Somebody may wonder about the in vitro mutation rate during cell culture. So we conducted this kind of analysis. So after the passage, we counted how many artificial mutations are acquired. So our analysis is if we culture around 1 month of a single cell, then there will be additional around 7% more mutations. So, or in absolute number, per day, around 10 mutations are to be acquired. So, although this is not very low, but compared to whole genome amplification, it will accumulate 1 million, for example, in the single whole genome amplification step. So, compared to the whole genome amplification, the clonal expansion method provide much cleaner the whole genome sequences. And for copy number changes, as you can see here, that it provides very clean data set. So we are very happy with the clonal expansion method. So let me show how we constructed the early phylogenies using somatic mutations. So from the donor body 3, the third the donors, we collected 43 clones. So for example, the first clone was derived from the left eye muscle, and from the clone whole genome sequencing, we identified around 2,000 somatic base substitutions. From the clone from the low pubic region, we identified around 1,400 base substitutions. And interestingly, 8 mutations are shared between those 2 clones. That means that it means that those 2 clones share a common ancestor, which accumulated 8 somatic mutations from the fertilized egg. We derived another clone from lower pubic region again. We identified 1,700 base substitutions, but interestingly, this clone shares no mutations with the previous 2 clones. So we can say this cell is the progeny of a completely different lineage. And from the fourth clone, which was derived from the right side fibroblast, we identified 1,300 mutations, and it shared another set of 8 mutations with the third clone. So those two and those twos are separated in terms of the early lineages. So we can continue this kind of analysis again and again for more clones, and we can generate this kind of the lineages by using of somatic mutations. And these are two clones are actually derived from the right keratocyte, that is sorry, I don't know why. So keratocyte, which is the ectodermal tissues, and the red clones were from the mesodermal tissues. So their common ancestral cells are actually the cells earlier than the gastrulation. So we can say these mutations are actually early embryo mutations, which were acquired prior to the gastrulation stage. This is more systematic analysis. So for example, from DB3, we derived 43 clones, and from each clones, we identified many somatic mutations. And actually, for the 30 clones out of 43, they shared 8 mutations, or set 1 mutation. And the remaining 13 clones have another set of 8 mutations, set 2 8 mutations. So every cell has either set 1 or set 2 mutations. No cell has both the mutations, and no cell has none of the mutations. So we concluded that those mutation sets are mutations acquired in the 2-cell stage embryo. So these mutation sets are accumulated in the first lineage, the first cell, in the 2-cell stage embryo, and this mutation set was accumulated in the second cell of the 2-cell stage embryo. So from around 100 the clone whole genome sequencing, we could construct this kind of early embryonic phylogenies. So from the 5 donors, we detected overall 480 the base substitutions and 49 indels as an early embryonic mutations. So those are one of the largest mutation set that are accumulating the early human embryogenesis. Sorry, I don't know why. And as you can see here, the early branchings, or early lineages were branched before 35 mutations by molecular time. So because mutations are like the molecular clock during the embryogenesis, we can measure each event by the number of mutation numbers, right? And the vast majority of the lineage branching was the lineage branching were actually before the 35 mutation by molecular time. Or in other words, 35 mutation by molecular time is our latest developmental lineage we can observe in our study. From the around 500 base substitutions, early embryonic base substitutions, we can observe the mutation signature. Most of the mutations are C to T, and many of them are T to C. And from the mutation signature analysis, we identify that our mutational spectrum can be explained by 2 dominant mutation signature already known and deposited in cosmic databases. So the first is signature 5, the second one signature 1. So the proportion of signature 5 is around 70%, and the proportion of signature 1 is around 25%. So those 2 mutation signatures are actually driven by endogenous mutation processes rather than the carcinogen exposure. So we can confirm that the early embryo mutations are acquired by errors of the DNA replication or some endogenous processes. From indels, we can do the similar analysis, and many of the indels are acquired by replication slippage in the homopolymer regions, or some unknown but endogenous signatures already deposited in cosmic. So we can also confirm here that indels are also acquired in early embryonic stage, and the mutation signatures are well explained by the endogenous mutational processes. From the mutations, we can calculate the mutation rate as well. So the interesting thing is the mutation rate for the first cell division and second and later cell divisions are different. So for example, the mutation rate for the first cell division is higher, around 4 per daughter cell per cell division. But across individuals, actually there are huge variations. So some individual has around 6, for example, DB3 has 6. The mutation rate is very high per cell division, and some DB6, for example, the mutation rate is quite low. So there is a huge individual variation, but the average is around 4 per cell division. And from the second cell division and later ones, the mutation rate becomes much lower, which is the 1.2 per cell division. So, so far I explained how we conducted the capture of early embryonic mutations, which was the capture phase. And from the 5 individuals, we confirmed that mutations were indeed accumulated from the first cell division, and from the clone whole genome sequencing, we identified around overall 500 early embryo mutations from the 5 individuals. Now, our phase, we are moving to the recapture phase. We can track those early embryo mutations in many different tissues of the same individual. So this is how we conducted the recapture phase. So from the same individual, we can collect many small bulk tissues, for example, from liver, from blood, or from skin, or whatever. So from many anatomical locations and organs, we can collect, we collected many different tissues. And those small tissues can be sequenced by many different approaches. For example, deep whole genome sequencing, for example, the blood was sequenced up to 100x whole genome sequencing. And some anatomical, some tissues like liver were later captured microdissected, the liver lobules were later captured microdissected and whole genome sequenced. And many skins or many different tissues were targeted. We trace the early embryo mutations by targeted deep sequencing. So by the understanding of their variant allele fraction of each mutation, we can understand the proportion of early embryonic cells in each tissues. So by the analyzing of those the proportion, we could trace the cell dynamics in early human embryogenesis. Let me show how we conducted that. So from the 5 individual bodies, we collected around 400 deep bulk tissues. And for example, in DB6, we collected around 143 bulk tissues from many different tissues like central vessels, heart, stomach, spleen, colon, intestine, or whatever. And so from 5 individuals, we collected around 400. And from all the individuals, the blood tissues were sequenced using 100x whole genome sequencing. And from the DB3, the hepatic lobules were laser micro dissected and whole genome sequenced. We can identify many interesting things from the recapture phase. The first one is the developmental contributions of the 2 earliest cells. 2 cells at the 2 cell stage embryo are unequal. So for example, this is the outcome from the capture phase of DB6, and this is the lineage 1, and this is the lineage 2. So there are actually 115 clones, and actually the contribution of each two earliest node will be around 50-50. That is the expectation from the uniform the distribution. But in this the phylogeny, we observed that the lineage 1 contributed around 86%, and lineage 2 contributed only 13%. So there is a huge asymmetry in terms of the contribution of those two earliest nodes. Those are confirmed in our recapture phase as well. So we sequenced 143 bulk tissues, and we measured the variant allele fraction of the lineage 1 mutations and lineage 2 mutations by targeted deep sequencing. As you can see here, in the vast majority of the tissues, the variant fraction of lineage 1 mutations are much higher than lineage 2 mutations. That means those progenies or cells from lineage 1 are much more dominant in the vast majority of the tissues in the same individual. In other words, one cell at a 2-cell stage embryo contributes more dominantly to the adult body in DB6. These are constant to the other 4 individuals. So as you can see here, we can find the unequal contribution in all the other individuals, although the relative contribution, the ratios are different across the individual. So this is the ratio is 1 and 2, and this is 40% and 60%. So that means from all the individuals, the unequal contributions are consistent, but the relative ratios are variable. That is what we observed from our capture and recapture phase sequencing. What is the plausible mechanism for the unequal contribution in every individual? This is what we think at the moment. And during the embryogenesis, a small fraction of cells are selected to the inner cell mass that will contribute to our adult body. And we believe that during the embryogenesis, there is such a bottleneck. There must be some bottleneck, and from the bottleneck, some kind of asymmetries are stochastically generated in the inner cell mass. If the lineage contributions are different in the inner cell mass, then that kind of asymmetry will be formed in all the tissues of the individual. So we believe that the asymmetry in the tissues are the reflection of the early cellular bottleneck only in embryogenesis. Next one is the clonal compositions in many different tissues. So for example, we can check or confirm the clonal composition of many different tissues. The first one is systematic differences between left and right tissues. For example, in DB6, as you can see here, this is the left and right tissues, and the clonal compositions are different across the left-right axis. Same, the findings are observed from DB6 as well. So this is a little bit complicated, but this is the interpretation. So there is a lineage 1 dominant lineage and lineage 2 in those individuals. Lineage 1 is dominantly contributing in whole body, but lineage 1 is more dominant in the right tissues. So that means the relative contribution of L1 and L2 are different across the left and right axis. So, and because it's the 2-cell at the 2-cell stage embryo, that means those 2 cells are not equivalent across the left and right tissues from the first cell division, the very earliest stage of the embryogenesis. So it means that there must be a physical barrier across the left and right axis from the early stage of the embryo. That should be first between the first and fourth cell division. The second asymmetry is the asymmetry between three germ layers. And for example, the blue lines are the variantary fraction in ectodermal tissues, and red lines are variantary fractions of mesodermal and endodermal is the yellow one. And from the around third to seventh cell generation, or from mutations that are acquired in this kind of early embryonic stage, it shows some asymmetry between ectoderm and mesoderm and endoderm. So systematically, variantary fraction of earlier embryonic mutation are systematically lower in ectodermal tissues. So it is again a little bit complicated, but the interpretation is ectodermal dominant lineage appeared from this stage, but were missed in the capture phase of our analysis because our phylogenetic trees were mostly derived from the mesodermal or endodermal tissues. So I don't want to go into much detail here, but we can identify that ectodermal tissues are diverging from third to seventh cell generation of an embryo. Also, we can identify many other things here. So mutations that are acquired in the 9th, 7th cell generation, a little bit later in embryogenesis, are to be confined in a specific anatomical position. So this is the clustering of many bulk tissues using somatic mutations, and they are the variant fraction. So this is a T-SNE, the clustering. And from this stage mutation, we can say these mutations are confined into the specific anatomical positions. For example, some mutations acquired at this stage are accumulated in the right upper extremity, or some mutations are only found in the stomach tissues, for example. So we can say tissues from anatomically close regions show similar mutation profiles of the mutations acquired as 9th to 17th cell generations. Or we can observe that some mutations emerge, some cells emerge at some specific time. For example, we can check the contribution of each clone in the phylogenetic tree in blood cell pool. So for example, this lineage is one of the lineages phylogenetic tree, but its contribution to blood is extremely high, up to around 50% in DB9. So that means this lineage is contributing more dominantly in blood tissue specifically. And this clone emerges at 8th cell generation in this individual. Or from the liver tissues, we can conduct the similar analysis, and thus one minor lineage in the phylogenetic tree is dominantly contributing to liver lobules. That means this lineage is not specifically, but dominantly contributed in liver genesis during embryogenesis. So this is the conceptual summary. So during embryogenesis, we can track each clone's contribution to each different tissues by somatic mutation, and we can observe the left tissue, right tissue imbalances from the first to fourth cell generation, and 3 germ layer asymmetry from 3rd to 7th cell generation, and from the regional imbalance from arm, kidney, liver, or whatever, from 6th to 17th cell generation. So this is a little bit tentative, but we believe that is the order of the events during our embryogenesis. Actually, there are so many mutations and so many tissues. So actually, it is not very easy to explain. from this talk. So for somebody who are interested in our data set, you may visit our browser, that is somatic clone viewer, and you can find all of our mutations and their tissues from this web-based browser. There were some unexpected observations as well. So the first one is mitochondrial DNA heteroplasmy. So in DB10, we derived 38 clones, and from 14 of them, we identified this mitochondria mutation. And this mitochondrial mutation was was present in almost homoplasmy in those 14 clones. And because their mutation is only found in this specific individual, it is unlikely that those mutations are acquired independently in each lineage. More plausible mechanism is the mutation was present in the fertilized egg of the embryo. And this is what the plausible scenario. So in the fertilized egg, there was the heteroplasmy of the mitochondrial variant. And during embryogenesis, the mutation can be fixed in some lineages, and they will be found as homoplasmy in the clone whole genome sequencing of our experiments. Or in some lineages, the mutation can be purified somehow, and in those lineages, we will not find the mutation in the clone whole genome sequencing. And when we estimated the levels of heteroplasmy in the fertilized egg, in DB10, in the fertilized egg, we estimate that the mutation was present in around 30% in the fertilized egg. And in the fertilized egg, although the levels are much lower, we found similar the conclusion. So all the fertilized egg, they carried some levels of mitochondrial heteroplasmy. And another expected observation is we found many chromosome level copy number changes, chromosome gain or chromosome loss in the clone whole genome sequencing. And the sex chromosomes are actually enriched. So for male clones, Y chromosomes were frequently missed. And for female clones, X chromosomes were frequently missed in the clones. So we believe that the sex chromosome loss can be frequent in our normal aging processes. So this is the tentative summary So the whole genome sequencing of single cells by using a that approach, we can reconstruct the early cellular phylogenesis of human development. And from our approach from 5 donors, we identified the mutation spectrum and mutation rate, and we identified the widespread unequal contribution of first 2 cells and unequal contribution of the lineages across left-right tissues, region layers, and specific tissues like liver and blood. And we can also observe the mitochondrial heteroplasmy present in the fertilized egg, the copy number changes in normal cells across the aging processes. And the paper was published last month in Nature. So what? So this is our next question. So for example, from the C. elegans, from the worm, transparent worm, the Dr. John Thurston identified the complete embryogenesis, the cellular dynamics in embryogenesis by just visual inspection. But this is impossible for human development. So what if? So in this project, we sequenced only several hundred clones from individuals. But what if we can sequence 1,000 cells, 10,000 cells, 100,000 cells, and all the cells from an individual body? Then, using the similar approach, we can finally the reconstruct the complete the developmental processes of the human embryogenesis. That will be possible. And if the genome sequencing cost drops to $10, then probably we can conduct this kind of analysis. So this project was actually conducted by collaboration of Gyeongbuk National University School of Medicine and Hospital Department of Anatomy, the Professor Ji-won Oh, who contributed many single cells from the postmortem tissues for us. And the sequencing and most of the bioinformatics analysis have been conducted by Dr. Sung-Yeol Park and Dr. Yul Kim in our laboratory. And we are deeply indebted to the individuals who donated their body for this study. Thank you very much for listening, and I'd like to stop here. Thank you.
Thank you very much, Young Seok. That was a very impressive tour of your work. We have one question in the chat. I have lots of questions, but I'll go to the question from Franziska Paul first. Could a lower mutation rate, like in the ectoderm, be explained by fewer cell divisions?
Okay, that's a great question. So that is what we believe is this is because of our technical limitation. So let me go to here. So we derived many single clones from individual body after warm autopsies, but not all the tissues are actually proliferating. So fibroblasts or the muscle cells, they are proliferating very well. So we can establish those clones relatively very easily. And also endodermal tissues like the gut cells, gut epithelial cells, can be also established relatively easily. But ectodermal cells were very difficult to establish as clones. So in our phylogenetic tree, I would say the branches to the ectodermal dominant lineages should be somehow missed in our phylogenetic tree. So that means those ectoderm specific mutations are missed in our the capture phase, right? So because it was not detected in the capture phase, it could not be confirmed in the recapture phase. So it was caused by our technical limitation, and to improve our phylogeny, then we needed to collect more ectodermal tissues from the capture phase.
Thank you very much. There's a question from one of our panelists, JJ Liu. Maybe you can just ask it verbally.
So,
JJ, you want to unmute yourself and ask your question?
So let me read the question, okay? So from Franziska, it would be super interesting to do this in identical twins to see just how deterministic the process is. Sure. So we are actually conducting the twin analysis as well, monozygotic twins. But the thing is, from the twins, we can collect blood tissues quite easily. And from the buccal swab, we can buccal epithelial cells. But most of twins are actually, they are healthy and they are live. So we cannot collect many different tissues or cells from many different anatomical locations. That is very, very difficult. It probably is almost impossible. Or those two twins should provide their bodies simultaneously or whatever. So from that kind of technical reason, it is not very easy to collect many cells from the twins. But from the buccal swab tissues, buccal epithelial cells and blood tissues, we are doing the similar the studies. But because we could not collect that many cells, the resolution is much lower. Okay.
Makes sense. Okay, I think while we're waiting for JJ, we have another question from Bruno. Extra embryonic tissues like placenta will need to be incorporated to have the full lineage. Do you have plans for that?
Yeah, so you are all very great question. You know what I am doing at the moment. So we are collecting placenta tissues from the twins. So for the postmortem studies, most of the people are aged person, right? So we could not, their placenta tissues are not left. Or some individuals of the placenta available, they are still living, right? They are the healthy persons. So it is very difficult to collect both the placenta and the many cells from the somatic cells. But if we can conduct this kind of analysis, it will be super. It will be fantastic. And also Peter Campbell in Sanger Institute, he recently conducted this kind of analysis from stillbirth samples. So they can collect the placenta extraembryonic tissues and stillbirth the baby tissues, and they conducted a similar analysis. So it was published this April or something. So you can find that paper published in Nature. And also from the twin, we are collecting also placenta as well. So from the twin studies, it can be this kind of analysis can be available.
Thank you, Young. So I think we're still trying to figure out how JJ can unmute himself. If one of the hosts can help us with that, that'll be great. In the meantime, one more question from Bruno. Any genomic loci or genes that are more likely to acquire somatic mutations?
That's another great question. So we can, some mutations are actually overlapping. So we don't find any driver mutations that are frequently found in cancer. We found some TP53 mutation from one or two clones, but those are I don't think they are pathogenic mutations. So most of them are passenger mutations, and many of them were not the overlap of each other. But in some clones, some mutations were actually overlapped. And if we analyze their mutational signature, those mutations turn out to be accumulated by UV-mediated mutagenesis. So UV ultraviolet light can mutate the thymidine dimer, the regions, right? And by chance, the UV-mediated mutagenesis can make that kind of overlapping mutations. So without that kind of small example of exceptions, most of the mutations and all the early embryo mutations were not the overlapping to each other.
Thank you. So, okay, I think JJ is finally, we managed to unmute him. Go ahead.
Thank you. A quick question. Based on your data, most cells are very remotely related, right? You know, they only share very early stage of the. Is it because your sample biasing, sampling bias? You only sample the very limited number of cells in the body.
So let me go to the
linear tree, right?
Yes.
Yeah,
so
it looks like.
Sorry. Go to. Okay.
Yeah.
So most of the lineages were branched in the early embryogenesis before 35 mutation by molecular time, because we in the body there are 40 trillion of cells, right? And we think we collected cells almost randomly from the anatomical positions. So it is unlikely to expect that they are actually branched more recently.
I see.
Yeah, but we find some examples. So for example, those two clones and those clones were branched in very later stage. So, but they are anatomically isolated from the neighbor regions from the vicinity. So we find, although there are some of the examples, most of the clones are actually diverged in the very early stage. And that is what we expected. So for doing that, we established many clones from many different anatomical locations. That is why we don't find that many clones derived from the same lineages.
Thank you.
Thank you.
So Young Seok, really, you're going to have to sample, like you said, trillion cells, something like that, to get the full lineage.
Yeah.
But I was going to ask a related question, which is, and you've shown it here very nicely in this figure, that your sensitivity goes, for the later mutations, your sensitivity goes down because you rely on sharing.
Yes.
And it's, and to get shared mutations that are late mutations, you just have to get lucky, or you have to sequence lots and lots of clones, lots of cells. But let's extrapolate that a bit earlier to the very earliest division. I remember you showed that the first cell division, from one cell to two cells, has a higher mutation rate than the second cell division. Could that also simply be a sensitivity issue, that the earlier the mutation, the greater your sensitivity?
Yes, that's true. So if we increase the number of clones, and we can find many early mutations, that's true. So, but for the second cell division, for example, because we have many clones, so for the A cell stage or 60 cell stage clones, we will miss many mutations, early mutation, because of the lack of the clones. But for second and third clones, we think we have the sufficient number of cells, right? Even though, although we feel that we have a sufficient number of cells, we do not find that many mutations. So the sensitivity issue must be there, but that we don't think that would be that critical for the second cell stage, for the third cell stage. So another explanation for this is that the embryo starts their own gene transcription from second or third cell stage. So from the two cell stage, they use transcripts inherited from the mother. So that means they cannot activate the proper DNA repair mechanism. That is my just interpretation. So that may also contribute a little bit something in this the mutation rate differences across the cell stages.
That makes a lot of sense. It's good to hear the mechanistic explanation. And yeah, I think I would agree with you. You have enough clones to detect mutations in the second cell division. So yeah, maybe your sensitivity should be fine there. But really good to know, just at each division, if you could estimate the sensitivity somehow from the data. But I have a slightly different question. So there are many cell types that people presume come from multiple lineages. Okay, so for example, plasmacytoid dendritic cells are believed to come from the lymphoid and myeloid side.
Yes.
And similarly, fibroblasts, particularly in tumors, but perhaps normal fibroblasts also, people believe they come from many different lineages, and then they become fibroblasts. Fibroblasts are very diverse and they're kind of mysterious.
Yes.
What are their developmental origins? So for cell types like that, are you seeing evidence of, or do you have enough clones to start to address that question?
No. So we have we are underpowered for that analysis, because most of our branching is prior to gastrulation. That means prior to the differentiation into 3' layers. So we can only explore the events before the gastrulation event. So for conducting that, to answer that kind of question, we need to sequence many single genomes from the same type of the cells. So for example, if we can whole genome sequence 100 dendritic cells together with some fibroblasts, for example, then we can answer that question. So it will be very interesting work that should be done in the future.
Got it. Yeah. I guess your next study, which will have even more clones, I'm sure, as the cost goes lower and technology gets more efficient. We're officially over time. I'm going to ask one last question. And so I was intrigued by the asymmetry you showed, right? So between the first cell division, the two lineages, lineage 1 and lineage 2, you found like a 6 or 7-fold asymmetry. Yeah, for this individual. For different individuals, it was much milder, like 2:1 asymmetry or something like that. And I wonder, you have 5 individuals already. So in a sense, you already have a probability distribution asymmetry. Okay, you can get the standard deviation from these 5 individuals. You can get the standard deviation of the asymmetry.
Yes.
Once you have the standard deviation of the asymmetry, the standard deviation of asymmetry is inversely proportional to the square root of the number of cells that are contributing to your
inner cell mass.
Inner cell mass, exactly,
yes.
the epiblast.
Yeah, that will be possible.
Can you use that to, essentially like in population genetics, can you use that, binomial probability distribution, standard deviation calculation to say, what is the effective population size of your epiblast or inner cell mass or whatever?
Right. Yeah, so that's true. So that is also what I feel we can do. So for actually 5 individuals, the number of individuals are too small. So we did not make very good number for that. But if we conduct the similar analysis for much more individuals, 10, 20, that is not very high, but probably with that data, we can answer that question. So in this study, we focused on the common parts between 5, the embryogenesis, but the embryogenesis should be the individual event, right? There must be very high levels of variation. So if we analyze more and more individuals to 20 or 30, then we can answer that kind of questions.
Looking forward to that. Okay, actually, I said we're out of time, but I'm going to keep asking questions if that's okay. And you can tell me when you need to leave. The other question is, you showed X chromosome loss, which I was surprised by. I mean, there are publications on mosaic loss of Y chromosome as people grow older. When you say X chromosome loss, you're talking about the whole X chromosome or pieces of it? Or?
Most of them are whole X chromosome. So we think must be the inactivated chromosome can be missed from the somatic cell. And there is a chance that the chromosome loss events may be cultural artifact. That is also a possible scenario. But some X chromosome loss are common in the specific lineage. So that means the X chromosome was, at least that X chromosome was missed in the individual life, during the individual life. So I don't know how frequent it is in our population, but my conclusion is that event is happening and we need more deeper investigation of that event. And it will be quite interesting.
I think this is really amazing, this figure, the amount of not just X and Y, but you're showing loss of other chromosomes as well. And this is in whole chromosomes. So I presume if whole chromosome loss is so common, can we say that structural variants, that are, huge, but still smaller than whole chromosome, like 10 megabases or something, 1 megabase, will be even more common somatically?
So frankly speaking, we analyzed the genomic rearrangement or copy number changes longer than 10 megabases, and that is quite not very rare. But we did not analyze copy number changes smaller than 10 megabases. So if somebody can analyze that kind of data set, it will be also very interesting. But the thing is, many of the events are confined to single clone. So it is not easy to distinguish that if they are cultural artifacts or they are actually present during the lifetime of the individual. That was the difficult thing.
I see. Okay, so they're not repeated. It's just one clone.
Yes.
Okay. That makes a big difference. So they're quite rare then. If they're real, even if they're real, they're quite rare.
Right. Compared to somatic base substitution and indels.
Okay. Well, we could go on talking, but I'm going to let you go. That was brilliant. I think more data and more findings in one paper, like normally people would even stretch all this out into 2 or 3 papers. So really impressive. Thank you so much for your talk, and hope to have more interactions, and hope to see you in Singapore.
Thank you. See you soon. Thank you, everybody.
Thank you, Young Seok. Thank you all.